what happens after an injury? Inflammation. So what is inflammation, that we know, but we need to know how the pain starts, the nociception starts, or the relationship between the inflammation and the nociception. When a tissue is injured, it is broken, so the cell walls are disrupted, and if the cell walls are disrupted, the phospholipid bilayer, those lipids, they are converted into prostaglandins. These prostaglandins are synthesized with the help of one enzyme we know, that is cyclooxygenase. Enzyme may be 1 or 2. And this prostaglandin, now if there is the post-synaptic, this is the receptor or the end of the nerves, the free nerve endings, which has many receptors here, many pores, many receptors. And also, this may be myelinated or unmyelinated. I am drawing it myelinated. There are also some branches like the dendrons also may be there. They are also can be myelinated, covered with the Schwann cells. Now what happens? These prostaglandins, they come to stimulate the special type of receptor, that is sodium NAV receptor. They stimulate this so that this membrane potential, the inside of the cell, nerve cell, which was more negative, it becomes a little bit positive. So that if it is become positive, then immediately there will be electric current generated. That we will discuss later on. But it can stimulate. It can stimulate the endings, nerve endings. How it stimulates this prostaglandin. This again, after this tissue injury, lots of protons or hydrogen ions, these are also liberated. These hydrogen ions and protons, they also stimulate. There are another type of maybe receptor, the temperature receptor TRPV1, that is also stimulated. And also it stimulates, there are some acid sensing channels, ion channels. They also stimulate these protons, acid sensing ion channels. And so what happens, through these the ions will enter, the sodiums will entering into the nerve cells to make it. We know to start the electrical current in the nerve, it needs to be positive. Initially it was negative, so all efforts are done to stimulate it. That means to generate positivity here, to push some positive ions inside. And similarly, during an inflammation, lots of other cell comes in, like we know the neutrophil will come. If the neutrophil comes, neutrophil liberates lots of interleukins alpha, 1 alpha. They also liberate TNF, T1 necrosis, TNF alpha. And so this interleukin and this TNF alpha, interleukin beta also are mostly secreted here, interleukin 1 beta. So interleukin, TNF, so these all lead to, they stimulate again those thermal channels. They also stimulate these thermal channels, and they also stimulate these acid sensing channels and other receptors in the presynaptic neuron. And then again you need there are other structures which come into play. What are those? There are macrophages, this mast cell. Mast cell also comes, and from the mast cell they liberate 5-hydroxytryptamine, and also they liberate histamine. So this histamine and 5-hydroxytryptamine both are liberated. Also this mast cell, they also stimulate with the platelet activating factor, the platelets. The platelets are there, and these platelets, they also liberate lots of 5-hydroxytryptamine. So both the mast cell and the platelet, they can liberate this 5-hydroxytryptamine, and the role of the 5-hydroxytryptamine is stimulate a special type of another cytokine that is very important, that is bradykinin. So this bradykinin, that also comes from the injured tissue. It can also come from the Schwann cells. It can also come from the keratocytes. So from lots of tissue and also from the plasma. This bradykinin is very important, a very important cytokine which stimulates the presynaptic membrane or the many receptors, like they are called these G-coupled receptors. These G-coupled receptors are stimulated by the bradykinin. So we know 5-hydroxytryptamine and histamine both also makes the bradykinin more powerful. They also sensitize this receptor so that bradykinin can act better. So 5-hydroxytryptamine and this histamine assists bradykinin. And also, so we can understand these acids, these protons, the prostaglandins, the bradykinins, 5-hydroxytryptamine, histamine, all come into play as chemical energies. These chemical energies, they are also from the platelet, comes also direct energy, ATP. It is a purine metabolites. And this ATP is also, they stimulate, there are special type of receptors, that is purine receptor, P2X receptor. So they also stimulate, and there are other cytokines which also come into play in case of a prolonged inflammation. In case this inflammation is there, so there are some other structures, some other cytokines which will come into play. What are those? We know the nerve growth factor. This nerve growth factor, basically they are released in the central nervous system, but it is prepared in the dorsal root cells also. And these nerve growth factors from the dorsal root to the antidromic direction, they can be liberated from the branches of the other nerves also. See, these nerve growth factors are also liberated. It comes from the central nervous system, and also it comes from the Schwann cells also. Schwann cells also can prepare this nerve growth factor. These nerve growth factors can be prepared by many cells, like this epidermal cells, like the platelets. So all can release lots of nerve growth factors, and which will again, they can stimulate these nerves, they can make them more sensitive, they can make upregulation of these receptors. So in a very short time, more receptors will be there, will be pushed to there. This nerve growth factor, they maintains the growth of the nerve so that the injury is repaired. So we can understand these chemical informations, these chemical energies by so much of cytokines coming from the so much of cells, starting from the neutrophil or the mast cell or the platelet or the epidermoid cells or the broken tissues, even from the Schwann cells. So these together, also there is temperature. This temperature also stimulates this temperature receptor TRPV1. So these receptors all together, they lowers the threshold. They make these inside the nerve cell more positive. And this is how the inflammation stimulates the pain nerve fibers, A, delta and C nerve fibers.